Hello everyone, welcome to my Biology 110 presentation. Today I'm going to talk about cell transformation and a little bit of synthetic biology. First, I will talk about the principle of cell transformation, which is conducted in Lab 6. Second, I'm going to talk about my real-world example of using the cell transformation is synthetic water pollution detected cells. So cell transformation is the process of transferring foreign DNA into the competent cells. The competent cells usually refer to the cells that have the ability to pick up foreign DNA from the surrounding environment. The foreign DNA here is usually referred to the plasmid. Normal cells cannot accept foreign DNA. Therefore, we need to make it into a competent cells through chemical way or physical way. The chemical way includes putting the cell into the salt solution and doing a heat cool-down shock. The physical way usually includes the electrical pulses. The plasmid is a small circular double-stranded DNA found in the bacteria, as we see here, that can replicate and transcript independently. The plasmid usually contains a region of replication, antibiotic resistant gene, selectable marker, and the inserted gene. So here is the process of cell transformation through chemical way and physical way, electrical operation. So basically is putting the plasmid into the cells. It is the same as what we did in the lab 6. We have a DH5 alpha competent cells. We insert the plasmid into the competent cells, which we have the transformed cells. And we use the ampicillin resistant gene and the LAC-Z gene to select the cells that is already transformed. The importance of the cell transformation is that the cell transformation is a tool to transform genes to a cell for us to do the gene testing or to produce specific protein. Now, the plasmid is very powerful because you can see here that there are two restriction sites in this plasmid. So it can be cut by a restriction enzyme. So we can change, actually, if it is cut, we can change the inserted gene in the plasmid and build a recombined plasmid and use the cell transformation, transform into the bacteria to see the performance of our inserted gene. Here is the graph showing the transformation process. We just cut the inserted gene, original gene, out of the plasmid, and we transformed and we recombined it with a new gene. Okay, now let's talk about my real-world connection, the synthetic water pollution detected cells. So here's the introduction of our project. You guys can refer to this webpage to see the detailed description of our project. Basically, what our project, by the way, it is a IGM competition project. So what we did is that we synthesized cells that can produce fluorescence when interacting with copper. Therefore, we can indicate the level of concentration of copper in water. The principle is that now we have a E. coli already transformed the cells. That means it already has the transformed plasmid. When the copper goes into the cell, it will deactivate the repressor combining with this operator, which is synthesized by machine. So the reporter gene, which is the fluorescence shown gene, will be expressed, showing the fluorescence. Therefore, we can use the level of fluorescence to indicate the level of copper. However, the problem is that we need the cell to be more sensitive. So we have to increase the cell's sensitivity to copper. What we did is that we keep synthesizing the operator gene using machines, changing the base pair of the genes. And then we transform it into the E. coli cells using the same transformation methods. And then we test its sensitivity of copper. we did a lot of testing. This is just only a part of it, and we have their sensitivity according to the copper concentration, and we found our best mutant. If you guys have interest, you can go to our website to see the results. Yeah, here's my reference list, and that's all. Thanks for your listening.